Pancreas are a glandular organ in the upper mid region. However, it fills in as two organs in one, a stomach-related exocrine organ and a hormone-delivering endocrine organ. A stomach-related exocrine organ and a hormone-delivering endocrine organ. It is a glandular organ. ఎక్కడ ఉంటుందంటే upper mid region. Okay. It fills in as two organs. ఒకటేంటి stomach related exocrine organ and a hormone delivering endocrine organ. The production of pancreatic hormones including insulin, somatostatin, gastrin and glucagon. They plays an important role in maintaining sugar and salt balance in our bodies. Okay. The most important hormone that the pancreas produces, insulin. Okay, and the pancreas has both, as earlier I said, both exocrine and endocrine functions. Okay. And, next, coming to the point, it working as an exocrine, as earlier we discussed, exocrine organ. The pancreas discharges catalysts to separate the proteins, lipids, sugars, and nucleic acids in nourishment. Okay, exocrine organ వచ్చేసినప్పటికీ pancreas ఏం చేస్తుందంటే discharges catalysts to separate the proteins, lipids, sugars, and nucleic acids. Okay, whereas in endocrine organ, pancreas secretes the hormones insulin and glucagon to control గ్లూకోజ్ లెవెల్స్. ప్యాంక్రియాస్ ఎండోక్రైన్ ఆర్గాన్కి వచ్చేసినప్పటికి ప్యాంక్రియాస్ ఏం చేస్తుందంటే హార్మోన్స్ ని సెక్రెట్ చేస్తుంది. ఏంటేంటి అంటే ఇన్సులిన్, గ్లూకోగాన్. ఫర్ వాట్ పర్పస్ మీన్స్ టు కంట్రోల్ గ్లూకోజ్ లెవెల్స్ ఫర్ ద డ్యూరేషన్ ఆఫ్ ద డే. ఓకే, both of these different capacities are essential to the body's endurance. Okay, both of these different capacities are essential to the body's endurance. These are created by a specific tissue in the pancreas. Okay, these both exocrine and endocrine are created by a specific tissue in the pancreas and afterward discharged to the slim framework and arrived at the liver by the intravenous dissemination. This is the overview of pancreas. Okay, venous dissemination. Okay, and the specific tissue is called islets of Langerhans. Islets of Langerhans speak to around 1 to 2% of the pancreas. Okay, speak to around 1 to 2% of pancreas. Three kinds of cells are recognized in these islets of Langerhans: A cells, B cells, D cells, and F cells. Okay, A cells responsible for glucagon production, that means 25% of all islet cells. B cells responsible for insulin production, that means 60% of all islet cells. Okay, and D cells are responsible for somatostatin production, that means 10% of all islets of cells. Okay, finally F cells are responsible for pancreatic polypeptide production. It is also 5% of all the islets of cells. This islets of Langerhans assumes an essential job in starch digestion, thus in a plasma glucose absorption. The islets of Langerhans ఏం చేస్తుందంటే it assumes starch digestion, thus in a plasma glucose absorption. It involves both glycolysis and glycogenesis and glycogenolysis, lipolysis, and lipogenesis. Okay, in glycolysis, anaerobic transformation of glucose into lactate. Anaerobic condition లో transform అవుతుందన్నమాట. ఎలాగంటే glucose into lactate. It takes place in RBCs, renal medulla, and skeletal muscles. This glycolysis mostly takes place in RBCs, renal medulla, and skeletal muscles. Okay, whereas glycogenesis, the synthesis of glycogen from glucose. Glycogenesis means synthesis of glycogen from glucose. Glucose is stored in liver or muscle in the form of glycogen, and this serves to maintain a constant plasma glucose concentration. Glycogenesis serves to maintain a constant plasma glucose concentration. Okay, next, glycogenolysis, the breakdown of glycogen to glucose. Gluconeogenesis means production of glucose from non-sugar molecules. Those are amino acids, lactate, glycerol, etc. Lipolysis means breakdown of triacyl glycerols into glycerol and free fatty acids. This is the lipolysis. Okay. Lipolysis means breakdown of triacyl glycerols into glycerol and free fatty acids. Whereas lipogenesis means synthesis of triacyl glycerols. Okay. The main function of pancreatic hormones are responsible for the storage of fat and glucose as glycogen after the meal. Pancreatic hormones యొక్క మెయిన్ ఫంక్షన్ ఏంటంటే it is responsible for the storage of fat and glucose as glycogen after the meal, after we have taken the meal. Okay, this enables the mobilization of energy. It enables the mobilization of energy reserves due to food deprivation, stress and physical activity. It maintains the constant plasma glucose concentration. It promotes the growth. See, this is the overview of pancreas, and this is the islets of Langerhans. It contains A cells, D cells, Isnar cells, beta cells, PP cells. Okay, pancreatic polypeptide cells. Okay, it is located outside of the stomach exocrine gland, on exocrine organ, and inside the see here and a hormone delivering endocrine gland. Okay, in normal condition లో pancreas ఏం చేస్తుందంటే this is glucose uptake into cells, ఇన్సులిన్ రిసెప్టర్ అనేది తీసుకుంటుంది. ఓకే, ఇక్కడ టైప్ 1 డయాబెటీస్ లో ఏం చేస్తుందంటే డిస్ట్రక్షన్ ఆఫ్ బీటా సెల్స్ అనేవి డిస్ట్రక్షన్ అవుతాయి. అప్పుడు ఇన్సులిన్ అనేది ప్రొడ్యూస్ అవ్వదు. అప్పుడు సైకిల్ అనేది జరగదు అనమాట. ఇన్సులిన్ రిసెప్టర్ ఇది. ఓకే, ఇన్సులిన్ గ్లూకోజ్ ఏం చేస్తుందంటే అప్టేక్ ఇంటూ సెల్స్. టైప్ 2 డయాబెటీస్ లో ఇన్సులిన్ ప్రొడక్షన్ అనేది తగ్గిపోతుంది. సో ఇన్సులిన్ ప్రొడక్షన్ అనేది జరగదు. గ్లూకోజ్ అప్టేక్ అనేది జరుగుతుంది అనమాట. ఓకే, అండ్ నెక్స్ట్ కమింగ్ టు ద పాయింట్, insulin structure is a peptide that contains an alpha chain 21 amino acids long linked to a 30 amino acid beta chain via two disulfide bridges. These disulfide bridges, the precursor to insulin is preproinsulin, which contains a signal sequence that is further removed in the endoplasmic reticulum, converting the precursor into its prohormone referred to as proinsulin. Okay, insulin is a peptide anamata. It contains alpha chain and beta chain. Alpha chain contains 21 amino acids. And these are long linked to a 30 amino acid beta chain via two disulfide bridges. Two disulfide bridges లో ఈ బీటా చైన్ అనేది లింక్ అయి ఉంటుంది అనమాట. The precursor to insulin is preproinsulin. It contains signal sequence. అది ఏం చేస్తుందంటే ఫర్దర్ రిమూవ్ ఎక్కడ అంటే ఎండోప్లాస్మిక్ రెటిక్యులం లో ఈ సిగ్నల్ సీక్వెన్స్ అనేది ఫర్దర్ గా రిమూవల్ అవుతుంది. రిమూవల్ అయ్యాక అది ఆ ప్రికర్సర్ ని ఎలా కన్వర్ట్ చేస్తుందంటే ప్రోహార్మోన్ గా. ఆ ప్రోహార్మోన్ ఏంటి? ప్రోఇన్సులిన్. Proinsulin is changed into insulin after the removal of carboxypeptide from prohormone. Okay, next coming to the point, regulation. Insulin is mainly secreted in a response to increases in the blood levels of glucose. Insulin is mainly secreted in response to increase the blood levels of glucose. Higher level of glucose causes that glucose enters the beta cells and is converted to glucose 6-phosphate. A higher levels of glucose causes glucose enters into beta cells and is converted into glucose 6-phosphate. This creates the cytosolic ATP and leads to a closure of ATP gated potassium channels, leading towards depolarization, which results in voltage gated calcium channels opening, and the level of cytosolic calcium rises and recruits exocytosis of insulin and reopening of potassium ion channels. Okay, higher ఈ గ్లూకోజ్ 6-phosphate కి కన్వర్ట్ అయ్యాక ఇది ఎక్కడ జరుగుతుందంటే అది ఏం చేస్తుందంటే సైటోజోలిక్ ఏటీపీ క్రియేట్ అయ్యి ఆ తర్వాత లీడ్స్ టు క్లోజర్ ఆఫ్ ఏటీపీ గేట్ ఉంటుంది కదా, అక్కడ ATP gated potassium channels అవి ఏం చేస్తాయంటే depolarization చేస్తాయి towards depolarization, which results in voltage gated calcium channels. Okay, channels opening, and the level of cytosolic calcium rises. Cytosolic calcium అనేది ఇంక్రీజ్ అవుతుంది అండ్ రిక్రీట్స్ ఫైనల్లీ ఎక్సోసైటోసిస్ ఆఫ్ ఇన్సులిన్. తర్వాత మళ్ళీ పొటాషియం ఛానల్స్ అనేవి ఓపెన్ అవుతాయి. ఇన్సులిన్ సెక్రేషన్ ఈ స్టిమ్యులేటెడ్ డ్యూరింగ్ డైజెషన్, ఓకే, via acetylcholine, vagus nerve, gastrin and secretin. Okay, next function, insulin has anabolic and lipogenic effects. Insulin has both anabolic and lipogenic effects. The storage of glucose in the liver is stimulated. లివర్ లో గ్లూకోజ్ స్టోరేజ్ అనేది స్టిమ్యులేట్ అవుతుంది అనమాట. అండ్ ఆల్సో యాక్టివేట్స్ ఎంజైమ్స్ టు ప్రమోట్ గ్లైకోలైసిస్ అండ్ గ్లైకోజెనిసిస్. It activates the enzymes to promote glycolysis and glycogenesis. In addition, it promotes the uptake and storage of amino acids. It promotes the uptake and storage of amino acids. Okay, the kind of proteins and promotes growth. Insulin also increases the amount of GLUT4. Okay, insulin also increases the amount of GLUT4. Okay, glucose transporters are present in skeletal myocytes. Glucose transporters are present in skeletal myocytes so that glucose can enter into the cell. Okay, glucose can move into the cell in two different ways. One is with sodium as a secondary active transport, and the other one is through glucose transporters, which is nothing but facilitated diffusion. ఒకటేమో సోడియం ద్వారా సెకండరీ యాక్టివ్ ట్రాన్స్పోర్ట్ కింద, అండ్ ఇంకోటి గ్లూకోజ్ ట్రాన్స్పోర్ట్స్ కింద మూవ్ అవుతాయి అన్నమాట. ఓకే, దీన్ని ఫెసిలిటేటెడ్ డిఫ్యూజన్ ద్వారా కూడా జరుగుతుంది. ఓకే, insulin affects many organs. It stimulates skeletal muscle fibers, okay, to take up glucose and change it into glycogen. And it takes up amino acids from the blood and change them into protein. It acts on liver cells. It's insulin stimulating them to take up glucose from the blood and change it into glycogen, while inhibiting enzymes production that is involved in breaking glycogen back down. Enzymes production. It inhibits the enzyme production which is involved in breaking glycogen back down, inhibiting gluconeogenesis. That is the conversion of fats and proteins into glucose. It acts on fat, nothing but adipose cells, to stimulate the uptake of glucose and the synthesis of fat. It acts on cells in the hypothalamus to reduce appetite. In such circumstances, insulin activates these effects by binding to the insulin receptor. In such circumstances, insulin activates these effects by binding to the insulin receptor, a transmembrane protein, which is embedded in the cell membrane of the reacting cells. Okay, to sum up, the end product of these reactions is capacity of the dissolvable supplements, which is retained from the digestive system into insoluble, which are not soluble in water, and vitality rich items such as glycogen, protein, fat, and a drop in the level of blood sugar, etc. Okay, next, coming to the point, Glucagon structure. Glucagon is a polypeptide which is derived from proglucagon. That proglucagon nothing but glycine. Okay, glucagon secretion is stimulated by amino acids arginine and alanine, from where means digested proteins, and furthermore by hypoglycemia because of physical exercise. From digested proteins దగ్గర నుంచి ఈ గ్లూకోగాన్ సెక్రెషన్ అనేది స్టిమ్యులేషన్ జరుగుతుంది. ఎక్కడ అంటే అమైనో ఏంటి గ్లూకోగాన్ సెక్రెషన్ అనేది స్టిమ్యులేట్ అయ్యి ఉంటుంది. దేని ద్వారా అంటే అమైనో యాసిడ్స్, అర్జినిన్, అలనైన్ ద్వారా. అవి ఎక్కడ అంటే డైజెస్టెడ్ ప్రోటీన్స్ దగ్గర అనమాట. ఈ తర్వాత హైపోగ్లైసిమియా కింద కన్వర్ట్ అవుతుంది. అది ఎందుకు అంటే because of physical exercise and sympathetic driving forces. Okay, the discharge is hindered by glucose. Our discharge is hindered by glucose, somatostatin, and high plasma concentrations of free fatty acids. The main function of Glucagon is mainly antagonizes insulin. The signal from the glucagon receptor is spread via cyclic adenosine monophosphate. Glucagon increases glycogenolysis in the liver. Glucagon increases the glycogenolysis in the liver and stimulates gluconeogenesis from lactate, protein degradation and lipolysis may take place. Mm, it also stimulates gluconeogenesis from lactate, protein degradation, and lipolysis. Okay, its main role is to maintain the regular concentration of glucose. Its main role is to maintain glucagon. Main role is to maintain the regular concentration of glucose between meals to ensure constant energy supply. Okay, between meals to ensure constant energy supply. Okay. Next, coming to the point, somatostatin. Somatostatin is released in response to higher plasma concentrations of glucose, okay, and arginine. Somatostatin is released in response to higher plasma concentrations of glucose and arginine through paracrine pathways, which inhibits the release of insulin and also the secretion of glucagon, which this somatostatin inhibits the release of insulin and also the secretion of glucagon. During the deficiency of glucose, this process does not occur due to the release of catecholamines. During the deficiency of glucose deficiency ఉన్నప్పుడు ఈ ప్రాసెస్ అనేది జరగదు. ఎందుకు అంటే catecholamines రిలీజ్ అవుతున్నందుకు. అది ఏం చేస్తుందంటే ఇన్హిబిట్స్ ద సెక్రేషన్ ఆఫ్ somatostatin. Somatostatin యొక్క సెక్రేషన్ ని ఇన్హిబిట్ చేస్తుంది, స్టాప్ చేస్తుంది. ఓకే. Next, coming to the point, pancreatic polypeptide. The F cells of islets secrete a 36 amino acid pancreatic polypeptide, which reduces appetite. Okay, the function of pancreatic polypeptide is to self-regulate pancreatic secretion activities. The main function of polypeptide is to self-regulate the pancreatic secretion activities, that is both endocrine and exocrine. It also has effects on hepatic glycogen levels and gastrointestinal secretions. Okay, it also has effects on hepatic glycogen levels and gastrointestinal secretions. Its secretion in humans is increased after a protein meal, fasting, exercise, and acute hypoglycemia, and is decreased by somatostatin and intravenous glucose. Okay, its secretion in humans is pancreatic polypeptide secretion in humans is increased after a protein meal. Protein meal tarvata, fasting tarvata, exercise dwara, and acute hypoglycemia, and is decreased by somatostatin and intravenous glucose. Okay. Next, coming to the point, skeletal muscle is the main shrink. Skeletal muscle is the main shrink for expelling abundant glucose from the blood. The skeletal muscle is main shrink, okay, for expelling abundance గ్లూకోజ్ ఫ్రమ్ ద ఎక్కువ గ్లూకోజ్ అనేది ఎక్స్పెల్ అవడం వలన ఎక్కడి నుంచి అంటే బ్లడ్ అండ్ చేంజింగ్ ఓవర్ ఇట్ ఇంటూ గ్లైకోజన్. ఓకే. నెక్స్ట్, కమింగ్ టు ద పాయింట్, ఇన్హెరిటెడ్ ఫార్మ్స్ ఆఫ్ డయాబెటీస్. A few instances of diabetes result from mutated genes acquired from one of the two parentages. Okay, few instances of diabetes result from mutated genes. Okay, from mutated genes which are acquired from one of the two parentages. For example, the mutant inheritable factor for one or another of the transcription factors needed for transcription of the insulin gene. Okay, the mutant inheritable factor for one or another of the transcription. Okay, another transcription factors which are needed for transcription of the insulin gene. Okay, it changes in one of the two duplicates of the quality encoding the insulin receptor. It changes in one of the two duplicates of the quality which is encoded in the insulin receptor. Okay, these patients for the most part have extra significant levels of causing insulin. These patients for the most part have extra significant levels of causing insulin. Okay, however, imperfect receptors, the mutated receptors. Okay, however, imperfect receptors. The mutated receptors are maybe unsuccessful to be articulated accurately at the cell surface. These mutated receptors are maybe unsuccessful to be articulated, which are accurately at the cell surface, or maybe unsuccessful to convey an operative sign to the inside of the cell. Okay. Next, coming to the point, a mutated variety of the gene encoding glucokinase. Mutated variety of the gene encodes glucokinase. Mutations in the inheritable factor coding part of potassium channels. Mutations in the inheritable factor లో ఈ mutations అనేవి కోడింగ్ పార్ట్ అన్నమాట. Coding part of potassium channels. Ekkada ante cell membrane of beta cells. The channels become unsuccessful to close approximately, making the cell become hyperpolarized. Channels అనేవి unsuccessful అయిపోతాయి. For what? Because means to close appropriately, making the cell become hyperpolarized and blocking insulin secretion. Insulin secretion ని blockage చేస్తుంది. Okay, changes in a few mitochondrial qualities which decrease insulin discharge by beta cells. A changes in a few mitochondrial qualities takes place which decrease insulin discharge by beta cells. These elements are acquired from the mother as just her mitochondria get by in the prepared egg. Okay, these elements are acquired from the mother as just her mitochondria get by in the prepared egg. While symptoms usually appear in childhood or adolescence, symptoms ఎక్కడ యూజువల్గా ఎక్కడ చూస్తామంటే చైల్డ్హుడ్ లో కానీ అడల్ట్స్ లో కానీ లేదంటే పేషెంట్స్ with acquired diabetes vary from most youngsters with T2D in having a history of diabetes in the family, that means hereditary, and not being obese. Okay. Type 2 diabetes అన్నమాట T2D అంటే. Okay. Amylin. Next, coming to the point, amylin. Amylin is a peptide of 37 amino acids which is also released by the beta cells of the pancreas. Okay, which is also released by the beta cells of the pancreas. Some of its actions are which it inhibits the secretion of glucagon, it slows the clearing of the stomach, it sends a signal of fullness into the brain. Amylin sends a signal of fullness into the brain. The entirety of its activities will in general increase actions like insulin, diminishing the degree of glucose in the blood. Okay, entirety of its activities will in general increases action like insulin and diminishing the degree of glucose in the blood. A manufactured type of amylin with modification is utilized in the treatment of type 2 diabetes. Okay, this is the overview of pancreatic hormones. Okay, thank you.